Alright folks, today we're going to talk about cellular respiration, and it's a fairly complicated procedure, but we're going to break it down so it's a little bit easier to understand. And of course, the main thing here is to remember that cellular respiration is how cells produce energy, and the energy type that they're going to produce is called ATP, as you can see right here. Okay, so the cell needs this ATP to help fuel cellular processes. Without ATP, it can't do anything from something like building proteins or sugars or whatever it needs to do in that sense to transporting things across the cell membrane. Every individual activity that the cell does that requires energy requires ATP in order for that to happen. Okay, so keep that in mind throughout this process, that the whole idea behind this process is to create that ATP. Cellular respiration involves three phases. There's glycolysis, the Krebs cycle, and oxidative, nice line, Mr. Monahan, phosphorylation. Okay, so those are the three kind of steps of cellular respiration. And we're going to talk about each one of those in more detail a little bit later on. But before we do that, let's talk a little bit about what goes on in these processes in general. Okay, so you start off with glucose, as you see here. That's what you start off with. Glycolysis is the first step. Okay, so glucose is changed. As you can see, there's a bunch of arrows indicating reactions that are occurring, and glucose is changed into something called pyruvate. Okay, pyruvate then, as you can see, enters the mitochondria. This is the mitochondria. I'll just do it like that. It's the mitochondria and goes through the Krebs cycle, and minimal amounts of ATP are produced during glycolysis in the Krebs cycle, and then we get to the end part here where oxygen is inputted, and that's where we get the vast majority of our ATP through oxidative phosphorylation. Now, in the process of making all this ATP, we're also, in glycolysis in the Krebs cycle, we're also making some other important parts that are going to play a role in creating the huge chunk of ATP that we get here. Notice this box here is quite a bit bigger than these ones because we're getting lots of ATP. Now overall, and if this works properly, we will get a total of 36 ATP. So as we go through these, you're going to break down your notes into three parts: glycolysis, the Krebs cycle, and oxidative phosphorylation. It's going to give you lots of detail about each of those. Okay, but please remember that it is important that you understand what goes in, what the reactants are, and what comes out, what the products are of each of these phases. Okay? And then if there's anything else that's important as we go through, I will, of course, mention that. Before we start our more in-depth look at glycolysis, we're going to just take a look at the mitochondria itself, which is where most of the magic happens for these reactions. Okay, so the mitochondria is built for power production, and here are a couple of pictures that I wanted to show you. First of all, this is just a graphic that shows you what the mitochondria is meant to look like, both on the outside and the inside. Okay, you can see that. And then here, this picture is actually really cool. It's a scanning electron microscope picture of what a real mitochondria looks like. Okay? So, there's a couple of parts that you need to be aware of. Obviously, there's an outer membrane. That's kind of this brown membrane that you see here. And there's also an inner membrane. Now, the folds, and I'll kind of get some color here. These folds here, like right here, like that, and like that, okay, those are where the oxidative phosphorylation occur. They're called the cristae. Okay, notice that these things out here are just kind of straight. Not as much happens in those areas, okay, but in these folds, the cristae of the mitochondria, sorry, is where most of the oxidative phosphorylation happens. Now, this area in here, you can see it's kind of like a liquid. That's called the matrix. Okay, and that's where the Krebs cycle takes place. Now, glycolysis, which is the one we're going to talk about here in a second, talked about all these. doesn't take place inside the mitochondria at all. And glycolysis is the most primitive stage of cellular respiration. And we know that because every single organism on the face of the planet, at least the ones that we know of anyway, all use glycolysis to produce a minimal amount of ATP. Okay? So glycolysis takes place outside of the mitochondria in the cytoplasm. So it takes place in this area out here. But then, of course, the products from glycolysis will move into the mitochondria, and that's where the majority of the stuff that's going to happens to make the ATP. Let's take a little bit closer a look at glycolysis, and then eventually we'll move on. We'll talk about both the Krebs cycle and oxidative phosphorylation. Keeping in mind, you guys need to know what goes in to each, the reactants, what comes out of each, and an overall summary of what happens for each. Okay, so here's where we're going to start to talk about glycolysis if you're taking notes. Okay, so glycolysis. We always say, just for our purposes, that glycolysis starts with a glucose molecule. Okay? And as you can see, glucose will go through a series of changes, reactions. Each one of these reactions, don't forget, would be catalyzed by different enzymes. Okay? Don't lose sight of that, because enzymes control every single chemical reaction that happens within the cell. So just don't lose sight of that. All right. So glucose has, as you can see here, 1, 2, 3, 4, 5, 6 carbons. Remember the chemical formula, C6, H12, O6. Okay, so these little balls here, these darker balls, are meant to represent the carbons. So there's 6 carbons in glucose. Now, at the end, what you end up with are 2 pyruvate molecules. Notice that they have 3 carbon atoms. So glucose, which has 6 carbons, is split into 2 3-carbon compounds called pyruvate, sometimes called pyruvic acid. Anything that ends in ATE can also end in IC space acid. So pyruvate ending in ATE can also be pyruvic space acid, pyruvic acid. So you'll see both of those, so don't get confused. All right, so it goes through a series of changes. Here's the good news, guys. You do not need to know any of these names. Shh. Don't need to know them. Not important for our intents and purposes. If you go on and you start to study biochemistry in university, whatever, then you'll need to know those probably. Right now, not so much. Okay, so glycolysis, if we go back to the statement I was just saying, there's 6 carbons in glucose, and we split it into 2 3-carbon compounds. So glycolysis quite literally means the splitting of glucose. Look what we did, we split it in half. Okay? So that's the overall of it. Let's get into the little more nitty-gritty details here. Okay, so what are the reactants of this? Okay, well, glucose is a reactant. Here we have glucose. So 1 glucose molecule. We also have, if you see here, see everywhere where there's something going like this with an arrow? Okay, that's also a reactant. Okay, so at the beginning, we start off with 2 ATP. So glucose plus 2 ATP to start with. And then let's keep going down. Anything with these arrows, remember, is being added. Okay, none of these other things are important for our reactants and products. Okay, so here we have a NAD+, okay, which is an electron acceptor. It's an oxidizing agent in that it removes electrons, okay? So NAD+ is an oxidizing agent, and it removes electrons. Notice that there are 1 on each side, 1 for each pyruvate. Okay, so that's a total of 2 NAD+ for reactants. Also notice down here, we have 2 plus 2 ADP. So a total of 4 ADP as a reactant. Okay, so far we have glucose plus 2 ATP plus 2 NAD+ plus 4 ADP. That's our overall equation so far. All right? Okay. let's go and pick out the products now. Okay? So here's, I'm going to just erase something really quickly here. I've got my reactants circled. I'm going to underline, maybe in a different color here. Pick a different color. I'll underline, maybe not, that's not the best color, probably black. I'll underline my products, okay? So my reactants are circled. I'm going to underline my products. Now, notice here, coming out of the arrow, those are going to be my products. So I have 2 ATP turning into 2 ADP. Okay, so remember, ATP is energy. So in order to energize and split glucose, which is what glycolysis does, I need a little bit of energy to start. Okay, that's where that comes in. All right, let's come down here. Oh, here's a product, NADH plus H plus. Okay, so there's 2 of those. Remember, these are electron acceptors. So why is this H plus hanging out? Well, it's attracted to these electrons that the NAD just picked up. Okay, so they kind of follow them. And so there's 2 of those. Now down here, notice we've made 2 ATP on each side for a total of 4 ATP. All right, and then finally, we make 2 pyruvates. Now let's sum it up at the bottom here. So we took glucose, we took our 2 ATP, we took our NAD pluses, and we turned the glucose into 2 pyruvate. There's some water there too. Remember, every time that you use a particular reaction, if I'm splitting a molecule, then I'm using hydrolysis, right, which is going to actually create some water. But actually, hydrolysis is going to use water. But in this case, because I'm actually taking in electrons, I'm actually producing water here, taking away some of the oxygens from the glucose as well to make water. Okay? Not important for this part anyway. Because I made 4 ATP, but I used 2 ATP, we say that there's a net gain of 2 ATP. Okay? And then, of course, there's also these 2 NADHs that are produced. Okay? So glucose plus 2 NAD plus yields 2 pyruvate, 2 ATP, and 2 NADH. That's my overall for glycolysis. Okay? Let's go back up here just really quickly, just talk about this. There's the energy investment phase. Why is it called that? Well, because I had to invest 2 ATP to energize glucose and split it. Okay? Down here, though, it's the energy payoff phase because I end up making more ATP than I invested in the first place, for a net total of 2 ATP. So you can break it down like that. Phase 1, energy investment. 2 ATP are used to energize and split glucose. Energize, split glucose. And then down here, in the formation of pyruvate, we make 4 ATP and 2 NADH. That's the energy payoff phase. In the energy payoff phase, we make 4 ATP and 2 NADH. Energy payoff in the formation of pyruvate. Okay, so that in a nutshell is glycolysis and what you need to know about it. Please make sure you bring your questions to class. I'll check your notes, make sure you've got everything you need, and we'll go from there. Thank you.